Then we will connect Professor Alison Smith from University of Cambridge. But before her speech, I'd like to share a few notes about her when we are trying to connect. Alison Smith is professor at plant biochemistry at the University of Cambridge. Her research interests are focused around the metabolism of plants, algae, and bacteria, in particular of vitamin and cofactors, and how this can be manipulated to produce high-value compounds for nutraceutical and therapeutic purposes. Alison has pioneered the use of synthetic biology approach to develop algal biotechnology platforms and made major contributions to our understanding of the role of vitamins in cellular processes in algae and microbial communities. She established the Algal Innovation Center sited in the Cambridge Botanic Garden in 2016, and she serves on a number of advisory groups in the algal biotechnology sector and is a member of the board of INB, an independent agricultural research institute in Cambridge. She is a fellow of the Royal Society of Biology, the Marine Biological Association of the UK, and Corpus Christi College, Cambridge. And we connect to her to listen her speech titled "How Insight Gained from Algal Metabolism and Genomics Can Be Leveraged to Support Algal Biotechnology." The floor is yours. Thank you.
Thank you very much for that introduction. Can you hear me okay? I can't see or hear anything, so
okay.
Can somebody tell me if everything's okay?
Yeah, we can hear you right now.
Perfect.
You can continue, thank you.
Okay, so let me share my screen. Good, can you see my slides now?
Yeah, it's okay.
So thank you very much for inviting me to give this talk, and apologies that I can't be with you. I'd also like to express my condolences for the terrible events that are happening in the as a result of the earthquakes in the south of Turkey. It must be very worrying for you. So what I'd like to do in this short presentation is to tell you a little bit about the work that we do in my laboratory in general. And I hope that it will whet your appetite for algal metabolism. I changed my title to be slightly shorter than the one I sent to Bharat. But basically, I'm going to try and tell you about how the work that we've done on algal metabolism, or understanding algal metabolism, how we can leverage that to do something useful with it. So the research focus in my group at the moment is basically three strands. So we're interested in looking at the metabolism of vitamins and cofactors. We look at the interactions between algae and bacteria at the molecular level. And we are using synthetic biology approaches to do metabolic engineering to make algal production platforms for high-value compounds. In addition, we have this Algal Innovation Center, which was mentioned in the introduction, in the Botanic Garden in Cambridge. And my colleague Payam Mashahi is going to tell you about how we use that in his talk tomorrow. So I'm not going to mention it at all. So what I'm going to do is to tell you a bit of hardcore metabolism here. So we're interested in B vitamins. These are water-soluble molecules. And on the right, you can see a metabolic map showing all the many central metabolic reactions in the cell. And it's estimated that over half of the enzymes require a cofactor for their activity, and many of these are derived from B vitamins. So B vitamins are very complex chemically, and they therefore offer mechanistic or catalytic activity that the amino acids in proteins are not able to deliver. They have important roles in regulating genes and in interactions with other organisms. And we've been looking at all three of those fundamental aspects, then trying to think how we might apply that knowledge to biotechnological applications. So here are structures of 3 of the vitamins. On the left here is cobalamin or vitamin B12, and you can see it doesn't matter if you're not familiar with chemical structures, you can just observe, I hope, that it's very complicated. This is vitamin B1 or thiamine. It was the first vitamin that was identified. And this is biotin or vitamin B7. So what I want to do is to focus on, I want to focus entirely in this talk on cobalamin, but actually similar aspects are relevant for these other 2 vitamins. So to start with, I thought it would be worth showing you how diverse algae are. So there's over 50,000 known species, and below is a simple schematic showing the relationship of these organisms to one another in the so-called eukaryotic tree of life. And down here on the bottom left, this is the opistheconts, and this is where the animals and the fungi are. These are the amoebozoa, and all these other groups are contain photosynthetic lineages, but actually there is non-photosynthetic ones as well. So these are the distribution of the perhaps well-known algal species that you might be familiar with. For example, Chlamydomonas, like we were just talking in the previous talk, Chlorella, Haematococcus, and so on. And these are all eukaryotic organisms, that is, they've got a nucleus where their DNA is. And down here we have the cyanobacteria, and cyanobacteria gave rise to the chloroplasts that you find in all of these species. But they are not, they're not eukaryotic algae. And so spirulina is a cyanobacterium down here, and this will become relevant later on. Now, one of the things that we were particularly taken with is when we started to look at vitamins in the algal lineages, we found that rather than being independent of certain vitamins, many different algae require an external supply of the vitamins. So they're like animals, not like plants. Plants don't need to be given vitamins, but many of these algal species do require the vitamins to be added to the medium if you're trying to cultivate them. So here is a sort of more zoomed in series of algal species. So Chlamydomonas reinhardtii up here. This is not dependent on vitamin B12, so you can grow it without B12, but its very close relative, Volvox carteri, does need B12. And you can see there's other examples here. Chlorella vulgaris doesn't need B12. In the diatoms, Phaeodactylum doesn't need B12, but Thalassiocyra does. This diatom here also needs thiamine. The important thing is that none of the non-requirers, the ones that don't need B12, they don't contain cobalamin, so it's not that they're making B12, it's just that their metabolism means that they don't need it. And we showed some years ago that the reason for this is the presence or absence of these 2 enzymes. So vitamin B12 is a cofactor for this enzyme methionine synthase, or MET-H. But there's another form of methionine synthase, which is called MET-E, and it doesn't use B12 as a cofactor. And there's a correlation between the organisms, the algae here, that are dependent or independent of B12, which is shown by the presence of the MET-E enzyme. So Chlamydomonas reinhardtii has got MET-E, so it's B12 independent, whereas Volvox carteri has a pseudogene for MET-E, and it's B12 dependent, so it's not independent. And we demonstrated this connection experimentally by generating mutants of MET-E by doing gene editing in this case. So both in Chlamydomonas and in Phaeodactylum, the wild types are not dependent on B12, but when we edit the MET-E gene to knock it out, you can see that they no longer grow without B12. So this is the wild type here, the wild type Chlamydomonas, the host strain, and then when we knock out the MET-E gene, it doesn't grow if the B12 is not present. And a similar pattern is shown for Phaeodactylum. Now, in both the wild type Chlamydomonas and Phaeodactylum, there are these two genes, MET-E and MET-H, and if you grow them in the absence of B12, they will just use the MET-E gene. But if you give them B12, they will use the MET-H gene, and they actually switch off the MET-E gene. So this is showing an RT-qPCR experiment where this is the transcript detected for MET-E in the absence of B12 in blue, and then in the presence of B12 in red. And you can see in both of these species, the gene is essentially switched off. But interestingly, in this green alga, Coccomyxa, this has only got MET-E, and the MET-E gene does not respond to B12. So we think that the explanation for why this gene has been lost in such a widespread way is because there's a reliable supply of B12 in their environments. Engineer microalgae to be production platforms for high-value compounds, such as these plant natural products. So forskolin is a heart medication, and it's derived from coleus, which is an ornamental herbaceous plant. But plants make very many natural products, but they often make them in combination, so it's quite difficult to isolate them in a pure form. And in addition, they often make them only under certain conditions and only in small amounts. So there's considerable interest in having production platforms where you can have a much more reliable and high level supply. So we started this work by trying to express or generate a production platform to produce mannol oxide, which is an intermediate in making forskolin. And you need two enzymes to be expressed at high levels in the microalgae. So this is work done in Phaeodactylum by Katrin Geisler as part of our EU-funded merit programme. And this is the construct that she used. It's a bit complicated, but you saw it before in the previous talk, same sort of idea. So this is the gene for one of the enzymes, and this is the gene for the other enzyme. Both of these are under the control of the MET-E promoter, and then this is the selectable marker in the middle, so we can identify the transformants. So Katrin introduced this construct into Phaeodactylum, selected on the basis of resistance to the antibiotic using this gene here, and then screened the cells to find those that were making mannol oxide, which is only possible if both of these genes are being expressed at reasonably high levels. And you can see here that there's a peak which corresponds to mannol oxide in one of the strains that she had identified. But because the gene is driven by the MET-E promoter, if she adds B12, then that peak disappears, showing that it's genuinely the result of the expression of these two genes. And so because it's a strong promoter and because we get high levels, it's now possible for her to do further manipulation of the strains. And just to show you how the sort of things that we can do. So this is pyramiding in additional genes into the genome. And you can see as we add additional ones, for example, so this is the pathway to make mannol oxide. If we add the gene for GGPPS, which is required to make the substrate for the enzymes, GGPP, or if we add the gene for the first enzyme of the pathway, as we increase the numbers of these genes, you can see we get an increased amount of mannol oxide. And this sort of approach that we're using enables us to consider how we can optimize the production of this compound, and then ultimately to introduce subsequent enzymes towards the production of forskolin. So that's the metabolic engineering application. And the whole work that we do in many different aspects is we use synthetic biology. That's using modular cloning with standard parts. We have collections of a number of parts such as promoters, like the MET-E promoter, terminators, targeting peptides to get the protein into the chloroplast or another organelle and so on. standardized workflows to identify the transformants. We can do gene editing, and we have ways to regulate transgene expression. So this is a very active part of the work in our lab, and many people have been involved in that. The other way in which we've used, or another way in which we've used this fundamental understanding, is to consider ways to enhance the nutritional content of food or feed. And one of the conundrums about why there are so many algae that require vitamin B12 to grow in the laboratory is that they presumably also require that in the environment, but the concentration of this vitamin in the environment is extremely low. So you can't grow algae in filtered seawater. However, you can grow it if there are bacteria there, and the bacteria are the ones that are making B12. So here is an experiment that we did a long time ago with a B12 dependent alga. If we grow it in a flask with B12, it grows very nicely, it's nice and green. If we grow it without B12, it doesn't grow at all. But we can substitute the addition of B12 by adding a soil bacterium, in this case Mesorhizobium lotti. The culture is very stable. The two organisms, the bacteria and the algae, grow in equilibrium. And the bacteria can't grow in this medium either. So that shows that they are receiving photosynthate from the algae, and this stabilizes the interaction. And so we think this is how algae obtain their vitamins from the natural environment, because they live in communities with many other organisms, including many bacteria. Now, when you look in algal supplements, and this is a sort of proxy for algal food, as Vita Verdello was talking about, then many of the algal supplements contain B12, and there are details on the packets for these supplements that show that there are high levels of B12. And here are two examples. So you can get tablets of spirulina or powder of spirulina, which is a cyanobacterium. Remember, that's not a eukaryote, and it makes B12 like other bacteria. And here is chlorella, which is a eukaryotic green alga. It doesn't make B12. Doesn't make B12, but instead it can take it up from commensal bacteria, as in this example here. So we were interested in trying to see whether the B12 content and composition of things such as these supplements was appropriate to supplement diets. And this is partly driven by work that we've been doing with colleagues in India, because in India, particularly because of the very high level of veganism, plants don't make B12, and so if you don't have a very rich diet in animal products, then you can have you're at risk of deficiency. So many Indians have very low or low serum B12, and spirulina supplements are widely consumed to try and combat that. So together with colleagues at the University of Kent, Martin Warren and Ranjan Yajnik in Pune in India, we did a dietary intervention study using chlorella as an alternative to spirulina. And we did a pilot study showing that within 12 weeks, there was an increase in B12 serum in the individuals who were given the chlorella bars here. So they don't look wonderfully appetizing, sort of green, but apparently they tasted nice. We were set to do a very large scale study with over 600 participants, and unfortunately COVID put paid to that, because now many Indians just take very high levels of B12 to prevent or ameliorate COVID symptoms, and so we can't actually do this experiment anymore, but it's very promising. So why were we interested in this? Well, this is because there are actually lots of forms of B12, and shown here are two of them. So this is cobalamin, and it doesn't matter what the structure is, but just look here, there's this structure here, which is called DMB, and cobalamin has this group in the molecule. Whereas in this compound, It's very, very similar to this one, with the exception of this group here, which is adenine. And this is known as pseudocobalamin. And pseudocobalamin is much less bioavailable to humans than cobalamin. So we don't take it up very well, if at all, through the system in our guts. Now, pseudocobalamin is not bioavailable to our algal mutants either. So they're like humans in terms of their preference for the different forms. And you can show this by looking at the growth of the alga in different concentrations of the compounds. So this is a complicated slide, but just look at this point here. So this is cobalamin. This is increasing amounts of cobalamin, and you can see where there's very little and it's sort of limiting. They don't grow very well. As you increase the amount of cobalamin, the cells grow. If you use the same concentration of pseudocobalamin, you need much higher amounts to see any growth at all. However, if you add DMB, which is that lower axial ligand, then the cells it's called remodeling, and then that enables the cells to grow as well as if they had been given cobalamin. So this is quite complicated, but the take-home message is that it's possible for us to distinguish in a mixture of compounds whether there's cobalamin present, because we do the assay simply, or the growth assay simply without DMB, or cobalamin and pseudocobalamin added together by adding DMB. And so this is work done by my student Ellen Harrison, and she showed that cyanobacteria like spirulina, this is its generic name for spirulina, Limnospira indica, and this is a lab strain of cyanobacteria. She showed that if you only measure the presence of B12 in the absence of DMB, so you're just looking for cobalamin, there's no detectable B12 presence at all. When you add DMB, you can see that there's now detectable B12 shown by the growth of this mutant. So in other words, the cyanobacteria here, they're only making pseudocobalamin. And she did the same experiment with health foods and supplements. And so this is, again, quite a complicated slide, but where it says C, that's a chlorella tablet. Where it says S, that's a spirulina tablet or powder. So in the chlorella case, you can always detect, when there's no DMB present, you can always detect, the green boxes here, some B12, meaning there's some cobalamin. If you add DMB, for C1 and C2, there's an increase. In other words, there's a bit of pseudocobalamin present. In C3, it's only cobalamin. But for the spirulina, which is S1, S2, and S3, there's only pseudocobalamin present. So whilst there are many other nutrients that are present in spirulina, including high levels of protein and iron, probably it's not a good source of B12 for humans. So to summarize, I think it's true to say algal metabolism is complex, but indeed metabolism is complex. But by studying the metabolism of one group of compounds in particular, and these are the B vitamins, it provides insight into how environment and cellular processes interact, how the algal auxotrophy almost certainly arose because there was B12 in the environment. This led to the loss of the MET-E gene, and therefore the cells are B12 dependent. But that means that they interact in the environment probably more tightly with their bacterial partners. And then we've used that knowledge to provide novel innovations for the exploitation of algae, both in metabolic engineering and in trying to increase nutritional benefits from these organisms. So it just remains for me to thank my group here. The metabolic engineering was done by Katrin Geisler and also by Payam Ashahi, who's speaking tomorrow. And then Ellen Harrison and Shelby Newsad were doing the work on the mutants. And thank you very much for listening.
Professor Alison, thank you for your lovely contribution. And if it is possible for you, I would like to ask the audience if there is a question from them.
Sure.
You can raise your hands.
Alison, I hope you can hear me okay.
Yes, I can.
Great. This is Berat. Thanks for your talk.
Hello, Berat.
This question is related to your D-terpenoid work. In Phaeodactylum, is it strictly non-mevalonate pathway, or do you see any metabolic flux loss to the mevalonate pathways?
I'm not certain that we have done the experiments to determine that directly. What the Phaeodactylum has the non-mevalonate pathway as well, and we have expressed an enzyme for a sesquiterpenoid, so a C15 terpenoid, which is made from FPP, which is present in the cytosol. We were only able to see production of the sesquiterpenoid when we were targeting the enzyme to the chloroplast, when we put the FPP synthase into the chloroplast. So in other words, I mean, that doesn't really say that there is export of IPP into the cytosol, but it does suggest that there isn't export or import of FPP into the chloroplast. I'm not certain if that answers your question, Berat, but...
Just a little bit, but of course it means that we need to look at that later. Yeah, thank you.
Yeah, I think basically I don't think it's any different from work done in plants. So if you want to engineer the C15s and C30 terpenoids, expressing the enzymes in the cytosol is most effective. If you want to look at the monoterpenes and the diterpenes and so on, expressing them in the chloroplasts is much better.
Right. Thank you so much.
Let me check the chat box if there's a question here. I think there is no more question. Thank you, Professor Alison, for your contribution. We appreciate it.
You're welcome.
Oh,
Goodbye.
sorry. I'm sorry. There's one more question.
Okay. Hello. Thank you for your presentation. My name is Müge. I was wondering that as far as I can see, you also work with non-model organisms as microalgae. And I wonder that do you face any challenges while doing transformation or things like that?
We really don't do genetic manipulation of anything other than Phaeodactylum and Chlamydomonas. We have tried in the past for a range of different species related to Chlamydomonas, and none of them are particularly straightforward or successful, I'd have to say. So I can't say that we have any real positive results on that. That's not to say it's not possible. It's just that we don't work on that area.
Thank you.
Okay, I think there is no more question. Thank you for your contribution, Professor Alison.
That's fine. Thank you very much, and enjoy the rest of your conference.
Thank you.